Hi everybody, today we're going to talk about intracellular signals and secondary messengers. So intracellular signals are going to be kind of the exception to the rule so far. Everything we've talked about, you have a signal molecule that cannot get through the cell membrane, and so it meets with the receptor protein that's actually embedded in the cell membrane. These intracellular signals are going to be signals that can pass through the cell membrane. They're going to meet up with a protein that's located in the cytoplasm. The example that we're going to look at are hormones. So here you have testosterone. It is passing through that plasma membrane. It is meeting with a protein receptor in the cytoplasm, and it's then being taken into the nucleus. And you can see that it is interacting with this DNA here. So you have an mRNA transcript that's been made. That mRNA can then leave the nucleus, and here it's out in the cytoplasm, and a new protein is being translated from that mRNA transcript. And so with this example, you had an activated protein that actually turns on genes within the nucleus. So again, most signals never enter the cell. Most signals will meet with the receptor at the cell membrane, and so this is sort of the exception to the rule, are these intracellular receptors. All right, we're going to move on to signal transduction pathways, and so these are going to be sort of used to amplify the signal that the cell is trying to send. And there's often going to be multiple steps using relay proteins that we're going to call protein kinases. Protein kinase is just going to be a general name for an enzyme that transfers an inorganic phosphate from an ATP to a protein. And so here we can see out here, we have the signal molecule meeting up with the receptor protein that's embedded in the cell membrane. When this happens, you get an activated relay molecule, and so you're going to have this inactive protein kinase activated, and that is going to sort of continue down, and we call this a phosphorylation cascade because it's cascading and things are being phosphorylated. You have the transfer of that inorganic phosphate to activate proteins on down the chain until you get your cellular response. So again, protein phosphorylation is anytime you have the addition of an inorganic phosphate to a protein, which is then going to activate or turn on that protein. And amplification, it's all about getting this signal stronger, and so the protein kinases are going to work in a cascade with each one being able to activate several other molecules. And so the result is that one signal may actually activate many different molecules because of these phosphorylation cascades. This diagram is just showing you how one molecule, so in this case the binding of epinephrine to a G protein linked receptor, is going to sort of start this phosphorylation cascade. So you've got the transduction, which is going to activate that G protein. That G protein is going to activate the adenylyl cyclase. Then you're going to make cyclic AMP, and then you're going to keep activating these protein kinases all the way until you have your cellular response. All right, now on to secondary messengers. So secondary messengers are small water-soluble. They're going to be non-protein molecules or ions that again help to pass on this signal. And so these are going to be able to spread really quickly by diffusion, and again, they're going to help activate some relay proteins to help get that cellular response. We're going to look at a couple examples. We will look at cyclic AMP, we'll look at calcium ions. The last one that we're going to look at is IP3, or inositol triphosphate. All right. So cyclic AMP is a form of adenosine monophosphate. It's made from ATP by that adenylyl cyclase that I mentioned earlier. This is a very short-lived molecule because it will get converted back to AMP. But while it's around, it's going to activate a bunch of different protein kinases. This is just showing you, here you've got your ATP molecule with your 3 phosphate groups, adenylyl cyclase. You lose 2 of the phosphate groups and you have this other phosphate group. You can see a new bond is formed. That's what makes it cyclic, and it only has the one phosphate, so that's the monophosphate part. Calcium ions are more widely used, and these are secondary messengers for both the G protein pathways and the tyrosine kinase receptor pathways that we've talked about. These work really well because of differences in concentration between extracellular and intracellular environments, and they're pretty widely used. So you've got plants, mussels, they're used by lots of different kinds of organisms. All right, the IP3 is a secondary messenger that is attached to phospholipids of the cell membrane, and they get sent to calcium channels on the endoplasmic reticulum. When they get to those calcium channels, they will bind to it and they will actually open up the calcium channel, so then the calcium ions flood into the cytoplasm from the endoplasmic reticulum. And so what we're looking at here is a G protein linked system on this side, or again, you could use the tyrosine kinase receptor system. With either one, you're going to have the signal molecule link up to the receptor. In this case, you're activating the G protein. The G protein is going to make the secondary messenger, in this case the IP3. Same thing is happening over here. The IP3 is going to make its way to the endoplasmic reticulum. Here we have a gated channel, so this channel is going to normally be closed, and it's going to keep the calcium ions inside the endoplasmic reticulum. When the IP3 gets here, though, it's going to bind to this calcium channel. It's going to open it up. Calcium will flood out and will interact with some of these proteins in the cytoplasm. And when that happens, you are going to activate different proteins and you will get a cellular response. Alright, so your lecture question for today is, how do secondary messengers affect cell signaling? So how is it that you think cells would be affected if they did not contain secondary messengers? Alright, thanks guys.